Let's dive into how scientists solve mysteries. Not about missing cookies, but about life itself. A biological problem is any question about living things that sparks curiosity and can be investigated scientifically. For example, why are bees disappearing? Or why are your plant's leaves turning yellow? The key is that the question must be specific and measurable, something science can actually test. Framing the right question is the first step in any scientific journey. And to solve these puzzles, scientists use a step-by-step guide called the biological method. When you're faced with a biological problem, whether it's understanding why a plant is wilting or how a disease spreads in a population, you can't simply rely on guesses or intuition. Science demands evidence, careful observation, and logical reasoning to uncover the truth behind what we see in the natural world. Instead of jumping to conclusions, scientists gather data, look for patterns, and use their findings to build a solid foundation for their ideas. This approach ensures that every answer is backed by proof, not just opinion. Across the globe, researchers follow the biological method, a step-by-step process designed to turn curiosity into knowledge. It starts with identifying a clear problem or question. Next, scientists make detailed observations, collecting as much information as possible. They then form a hypothesis, which is an educated guess that explains what they've observed. From there, they make predictions about what should happen if their hypothesis is correct, and design experiments to test those predictions. After conducting experiments, scientists carefully analyze the results, looking for evidence that supports or challenges their hypothesis. This analysis often involves reviewing data, using computers to spot trends, and sometimes repeating experiments to be sure of the findings. One of the strengths of the biological method is that it encourages scientists everywhere to repeat experiments. By replicating studies in different labs and countries, researchers can confirm results and build a global understanding of biology. This process helps eliminate errors and ensures that discoveries are reliable and trustworthy. The biological method isn't a strict set of rules. It's more like a flexible toolkit. Scientists can adapt their approach depending on the problem, using creativity and teamwork to find the best solutions. This flexibility is what makes science so powerful and dynamic. Sometimes, experiments don't go as planned, and unexpected results lead to entirely new questions. This is a normal part of science, turning every answer into the start of a new investigation. The biological method keeps the cycle of discovery going, making science an ongoing journey of learning and exploration. Thanks to this method, humanity has made incredible breakthroughs in fields like medicine, genetics, and conservation. From developing life-saving vaccines to protecting endangered species, the biological method is at the heart of every major discovery in biology. Every scientific discovery begins with a moment of curiosity, spotting a biological problem that stands out from the ordinary. It might be something subtle, like a plant that isn't growing as well as its neighbors, or something dramatic, like a sudden change in color or shape. This first step is all about noticing that something is different and asking yourself, why is this happening? For example, imagine you're in a garden and you notice that some tomato plants are thriving, while others look wilted or stunted. This difference is your starting point, a clue that there's a problem worth investigating. The next step is careful observation. Scientists gather clues using both their senses and scientific tools. Qualitative observations involve what you can see, smell, or feel, like the color of leaves or the texture of the soil. Quantitative observations involve measurements, such as the height of the plants or the number of tomatoes produced. For instance, you might notice that the healthiest plants have leaves that are a deep, rich green, while the struggling ones have pale or yellowing leaves. This visual clue can tell you a lot about the plant's health. You could also use a light meter to measure how much sunlight each plant receives, discovering that the healthiest plants are in the sunniest spots. To dig deeper, you might collect soil samples and test them in a lab, checking for differences in nutrients or moisture levels between healthy and unhealthy plants. Counting the number of tomatoes on each plant gives you another piece of the puzzle, helping you see which plants are most productive. By combining both qualitative and quantitative observations, you start to build a complete picture of what's happening. This thorough approach helps you avoid missing important details. As you gather more data, look for patterns. Maybe the healthiest plants get more sunlight, have better soil, or receive more water. These patterns are the clues that guide your investigation. All these careful observations and measurements become the foundation for your next step, forming a hypothesis, a testable explanation for what you've observed. Without this detailed process of observation and data collection, any guess about what's wrong would be just that, a guess. But with solid evidence, you're ready to move forward with confidence. At this stage, it's time to take all the clues and observations you've gathered and use them to form a hypothesis. A hypothesis is more than just a guess. It's a thoughtful, testable explanation for what you've noticed happening. Scientists use their background knowledge, previous research, and careful observation to come up with a hypothesis that can actually be tested through experiments. For example, let's say you've noticed that some tomato plants seem to grow taller when they get more sunlight. You might form the hypothesis: plants grow taller because they get more sunlight. This statement gives you a clear idea to investigate. A strong hypothesis is always specific and testable. It clearly states the relationship between two or more variables, in this case, the amount of sunlight and the height of the tomato plants. This clarity helps you design an experiment that can actually prove or disprove your idea. Once you have your hypothesis, the next step is to make a prediction. A prediction is a statement about what you think will happen if your hypothesis is correct. For our tomato plants, you might predict, if tomato plants get more sunlight, then they'll grow taller than those with less. This if-then-if statement is important because it guides your experiment. It tells you exactly what to look for and what results you expect to see if your hypothesis is right. The prediction connects your idea to a measurable outcome. It helps you decide what data to collect and how to measure success or failure in your experiment. With a clear prediction in place, you know exactly what to observe and record during your experiment. This focus makes your investigation more organized and meaningful. Now you're ready to put your hypothesis to the test and see if your prediction holds true. This is where the real excitement of scientific discovery begins. It's time for action. Designing an experiment to test your prediction. Set up a controlled experiment. Change only one variable, the independent variable, like sunlight, and keep everything else the same. Use two groups. One gets more sunlight, experimental. The other, less, control. Keep soil, water, and other conditions identical for both groups. This way, any difference in growth is likely due to sunlight. Collect data by measuring plant height and other features over time. Careful data collection is your evidence. Now let's see what the results reveal. With your data collected, it's time to analyze and interpret the results. Organize your data in tables or graphs to spot patterns. If the sunlit plants grew taller, your hypothesis is supported. If not, it's refuted. Either outcome is valuable. Science learns from both. Share your findings so others can learn and repeat your experiment. Summarize your conclusion. Was your hypothesis supported or not? Each experiment, win or lose, leads to new questions and discoveries. One experiment isn't enough to declare a scientific fact. Repetition and confirmation by others build confidence in an idea. If a hypothesis survives many tests, it can become a scientific theory, a broad explanation backed by lots of evidence. Theories explain why things happen, like evolution by natural selection. Scientific laws, on the other hand, describe what happens often as a rule or formula, like Mendel's law of inheritance. Laws describe patterns, theories explain them. Both are essential for understanding the natural world.